Lymphatic system. Lymphatic system exists in all organs with the exception of central nervous system and cornea. Lymph is a tissue fluid that enters lymphatic vessels and then drains into the venous blood via thoracic duct and right lymphatic duct. Lymph contains clotting factor and it clots on standing in vitro. It is a transparent, yellowish in color, faintly alkaline, and its colloidal osmotic pressure is less than that of plasma. Its formation. Its formation is based on the transcapillary exchange. Lymph is derived from the interstitial fluid that flows into the lymphatics. It enters terminal lymphatics, which has the same composition of that interstitial fluid. The protein concentration of the lymph is uh, clear and 2 grams per deciliter, which is average protein concentration of interstitial fluid. Even large particles such as bacteria can push their way between the endothelial cells of lymphatic capillaries, thereby they can enter into the lymph. As we told, the formation of the lymph, it depends on the Starling forces, that is the rate of filtration at any point along the capillary depends on a balance of the forces which are called as Starling forces. Hence, fluid movement depends on the constant factor and the difference in the hydrostatic pressure minus difference in the osmotic pressures in the capillary and in the interstitium. Here K is the capillary filtration coefficient, Pc is capillary hydrostatic pressure, Pi is interstitial hydrostatic pressure, Pi C is capillary colloidal osmotic pressure, Pi I is interstitial colloidal osmotic pressure, which is negligible. And so this is the hydrostatic pressure gradient and this is osmotic pressure gradient. The total estimated lymph flow is around 120 ml per hour or 2 to 3 liter per liter per day. So this is how these are the factors which are affecting the Starling forces. They are going to differ at the arteriolar end compared to the venular end. The hydrostatic pressure at the arteriolar end is 37 mm of Hg and the osmotic pressure is constant. It is around 25 mm of Hg. Okay. So the fluid which is flowing across the arteriolar end, the pressure gradient is, since the capillary hydrostatic pressure is more than the osmotic pressure in the capillaries, the gradient is produces 12 mm of Hg across the arteriolar end, which flows from the artery into the interstitium. Whereas at the venous end, the hydrostatic pressure gradually reduces down and it comes down to 15 mm of Hg, whereas the osmotic pressure is going to remain at 25 mm Hg. So the pressure gradient created here is around 10 mm of Hg, and which helps in the drawing of the blood from the interstitium back into the capillary. So hence the fluid is absorbed here and the arteriolar end, it is going to be filtrated. So, and you can see the 12 mm of Hg gradient is created at the arteriolar end and 10 mm of Hg gradient is created while the fluid is being absorbed. Some amount of the fluid is going to remain in the interstitium and that is going to be drained by the lymphatics. Some of the functions of lymph. The lymph returns proteins, water and electrolytes from tissue spaces to the blood and thus controls the concentration of proteins in the interstitial fluid, and it is also going to control the volume of interstitial fluid and the pressure. The absorption, it helps in the absorption of nutrients, especially fats, from the GIT. Lymph acts as a transport mechanism to remove red blood cells that have lost into the tissues as a result of hemorrhage. And the lymph nodes act as efficient filters. They have sinuses lined with phagocytic cells that engulf bacteria, red blood cells, and other particulate matter, that is, bacteria or toxins, which are carried through the lymphs. They have a nutritive value, that is, it supplies nutrition and oxygen to the parts where blood cannot reach. Some of the applied aspect related to the lymph, that is hypoproteinemia, it it leads to decrease in the colloidal osmotic pressure. Therefore, the increased filtration occurs at the arteriolar end and decrease in absorption of the fluid at the venous end, resulting in abnormal collection of the fluid in the interstitial spaces, which is called as